Hello, my name is Ari Williams. I'm an MS3 at the IU School of Medicine. I worked with Dr. Maria Macon of the Purdue University Department of Biomedical Engineering. Together, we worked to decode pupil size from the neural activity in the mouse visual cortex. That is, we tried to convert the real-time neural signals of the visual cortex into a physical behavior. And we tried to create a model for these predictions, to translate the neural signals into an expected physical behavior, which we could then measure and determine the difference between our predictions and the measured actual value. Why did we do this? Well, brain-machine interfaces, or BMIs, aim to connect human brains to prosthetic devices, be it motor or sensory. These will require electrical signals of the brain, i.e. the commands of the brain, to be converted into a physical behavior using electrical signals. The visual cortex is the primary information processing unit of visual inputs in mammals. So the methods that we used an open source imaging data set from the Allen Institute of Brain Science. The experimental setup can be seen in the method's graphical summary. Mice were transgenically bred to have neurons which fluoresced when active. These mice were then placed and restrained in an experimental setup. Their brains were surgically exposed and imaged, as well as a range of physiologic behaviors such as pupil size, eye direction, head direction, running on a controlled ball, and a wide variety of other physiologic behaviors, as well as the neural activity. This can be seen in the neural activity imaging 2-photon calcium. This is an array of 128 cells over time. The activity of the cells is measured in fluorescence, with the most yellow being the most active. We took this data array and we smoothed the data using a filter, the Savitsky-Golay filter. Then we generated a linear regression model using the first 80% of the data. We used the first 80% of the data to generate this model, and then we tested the model on the final 20% of the data using the same y equals mx plus b format. The x value is the fluorescence of the cell. We used that to generate a model because we had the y value in the pupil size. Having the y and the x value meant that we could compute our m and b values. And we did this for the first 80% of the data, and then we used this new model to predict the final 20% of the data, which was separate. A correlation coefficient was computed, target cells of interest were pulled out and analyzed. For the results, the correlation coefficient was 0.376, indicating a moderate linear relationship between our pupil predictions and the measured pupil size. You can see the linear regression model on the upper middle of 3 graphs. The top is the raw data of pupil size over time. It was recorded from the mouse in the controlled experiment. This is right below it, the data that was applied using the Savitsky-Golay filter to smoothen the data out and remove any artifacts from the data gathering process. Below that is our linear regression model. And you can see when comparing the smoothened data versus our model data that the peaks line up fairly accurately, showing that we could predict spikes in pupil size based on the neural activity. But this wasn't enough because we wanted to actually make predictions on data which had never been seen by our model. And this is seen in the linear regressions prediction section of my poster. We used the first 80% of the data to generate the model, and then had the model make predictions on the final 20%, which we were able to compare to the actual real-world values. And you can see a great relationship of peaks lining up very well. You can see when there was a observed increase in pupil area, that our model also predicted a increase in pupil area, showing that we could measure brain activity and make predictions on the pupil size. And when we tested it by measuring the actual pupil size, it came out fairly accurately. But why does this help? Well, as I said, brain-machine interfaces seek to connect the brain to prosthetic devices, particularly damaged brains in patients. Immediate uses of this could be for patients with peripheral nerve palsies, which lead to inaccurate pupil sizes in patients. Various people suffer from either improperly dilated or constricted pupils and are unable to send brain impulses to the ganglia of the eye. Additionally, BMI has a wide variety of other uses, which we hope to investigate further to generate more useful therapeutic prosthetic devices for our patients.